Dear colleagues, this webinar is divided in two parts. The first is an introduction to the topic. The second focuses on the diagnostic approach to ocular surface disorders and dry eye. So let's talk about tears. There is so much more to them than you think. When we think of tears, we must consider their production, so the lacrimal functional unit, the composition, so all components of the tear film, the distribution, so the eyelids, the stability, so the surface, ocular surface epithelia, but also microenvironmental conditioning factors. The ocular surface, to me, is like a potential battlefield, and the main players are different. The lacrimal functional unit that includes all components of the ocular surface involved in the pathophysiology of dry eye. The microenvironment, I mean all factors continuously interacting with the lacrimal functional unit. It means ambient agent, microbiome, immune cells, hormones. The lacrimal functional unit is an integrated system comprising the ocular surface with sensory nerve endings, the interconnecting afferent innervation to the pons, where also emotional stimuli may have an effect, the efferent innervation to stimulate lacrimal glands, meibomian glands, and conjunctival goblet cells. The tear film is a two-layer dynamic unit functionally integrated with the ocular surface epithelial, and we consider a lipid layer of polar and non-polar lipids in surface, an hydrated mucin gel layer, and mucins are secreted by conjunctiva goblet cells. Their concentration decreases with distance from the epithelial cell surface. And then the corneal and conjunctival epithelia with transmembrane mucins in the glycocalyx of the most superficial cell layer to maintain epithelial wettability. This is a more detailed representation of transmembrane mucins in the glycocalyx of epithelial cells. They maintain ocular surface wettability because secreted mucins adhere to the arboriform structure. The ocular surface microenvironment defines the crowd of perfectly balanced elements on the ocular surface. A disease process may trigger a fight on the lacrimal functional unit battlefield with loss of balance. There is continuous interaction among the different tear film components. When one is defective, the ocular surface homeostasis is lost. Diagnostic tests help us to make the right diagnosis, and so we must examine all components. The lipids, and so with the meibomian glands, the tear fluid, so the lacrimal glands, mucins, the conjunctiva goblet cells, the proteins, so lacrimal glands and conjunctiva, but also the ocular surface epithelia with cornea, conjunctiva, and nerve supply, and the eyelids for blinking and ocular surface protection. But then we must consider ambient agent, microbiome, immune cells, hormones. Vision starts at the tear level. This is a very important sentence. This is very true for humans, but it is true also for animals. The tear corneal epithelial complex is the major light reflecting surface of the eye, accounting for approximately 65% of the optical power of the eye. A smooth and stable tear layer is essential for maintaining high-quality vision between blinks. So let's consider all components of the tear film, starting from the lipids. Polar lipids are stable. The polar heads are oriented perpendicularly to the aqueous layer. The hydrophobic tails are immersed in the nonpolar lipid sublayer. In case of lipid layer composition disorders, the lipid layer becomes unstable, collapse, and form lipid droplets with increased evaporation of the aqueous layer. And so we have evaporative dry eye. Meibomian glands secrete polar and non-polar lipids. In some cases, meibomian glands are visible under the transparent conjunctiva, but the best way to perform a careful examination is infrared meibography. In case of lipid layer disorders, qualitative and quantitative alteration of the lipid layer increases surface tension and evaporation, and so we have evaporative dry eye. The pictures show cases of blepharitis, expression of meibomian glands with altered meibum, and infrared meibography in meibomian gland dysfunction. What's about the liquid phase? The aqueous phase is a mucin gel loaded with water, proteins, electrolytes, glucose, urea, leukocytes, soluble mucins. There are multiple ducts, openings to drain tears secreted by the lacrimal glands. And a decreased tear production leads to aqueous deficient dry eye. When it is complicated by epithelial defects, there is loss of superficial wettability and also evaporative dry eye. Mucins are most important. Mucins have several roles: hydration of the ocular surface, corneal wetting, lubrication during the blink. They prevent pathogens from binding to the ocular surface. Mucins are secreted by the conjunctiva goblet cell and enter the aqueous phase to produce the tear mucin gel. When conjunctiva goblet cells are altered, the secretory mucin component of the mucin gel is affected, and so there is tear film instability and evaporative dry eye. Also, proteins have strategic roles for tear film. Tear film proteins derive from the lacrimal glands and via leakage from conjunctiva goblet cells. Almost 1,800 proteins constitute the human tear proteome, and the three main proteins are lysozyme, lactoferrin, and lipocalin. Lipocalin has many tasks. One is to be a carrier to transfer polar lipids and decrease tear film surface tension. Some proteins are potential biomarkers with important diagnostic role. In case of tear film protein disorders, there is loss of antimicrobial and anti-inflammatory defense of the ocular surface. We must carefully examine the ocular surface epithelia and their innervation also. The ocular surface epithelia are exposed to the external environment, but have tight intercellular junction that resist the entry of noxious pathogens. Also, the transmembrane mucin prevent penetrance of pathogens into the eye. Transmembrane mucins maintain epithelial wettability. If membrane-associated mucins have glycosylation defects, secreted mucin don't accumulate on the hydrophobic epithelial surface, and so we have evaporative dry eye. Sometimes, without a careful examination, the ocular surface may appear normal. In this cat, ocular surface topography lets see abnormalities we didn't suspect. The consequence is loss of wettability, evaporative dry eye, and a wet eye because of increased tear secretion. Sometimes ocular surface defects are easily diagnosed at the first approach. In some cases, we need to use stains like fluorescein or rose bengal, but it may be necessary to apply more advanced diagnostic tests like ocular surface topography. Preservatives, vehicles, and some drugs Use for ophthalmic drops may have toxic effects and alter the ocular surface epithelia with loss of cells and tear film instability. And ocular surface innervation has a role on tear production and distribution. The ocular surface nerve fiber derive from the branches of the trigeminal nerve and maintain the action of blinking and the tear reflex. The majority of the sensory nerve fiber are polymodal nociceptors activated by mechanical energy or noxious agents. At ocular surface stable temperature of 34 to 35 degrees, cold thermoreceptors continuously generate action potentials, and a moderate cooling, like for tear film evaporation, increases their activity, and so we have blinking. We must carefully examine also the eyelids. Early identification and management of eyelid abnormalities is mandatory to improve tear film quality and prevent evaporative dry eye. We must focus on blink quality and rate to see if there is a complete or incomplete blinking, and eyelids shape and position, but also presence of distichiae, ectopic cilia, and trichiasis, and then meibomian glands abnormalities and meibomian gland dysfunction, like shown in meibography here. Periodic and complete blinking spreads the film over the ocular surface and facilitates clearance into the nasolacrimal drainage system, so there is tear film turnover. Lack of eyelids closure is a common cause of exposure keratopathy in humans and also in animals. And in humans, the inability to close the eyes during sleeping is a cause of nocturnal lagophthalmos. Eyelids have important protective functions. Blinking defects, trichiasis, dystichiasis, entropion, ectropion, dermoids are common causes of ocular surface disorders and evaporative dry eye. We can't forget to consider also individual and external factors influencing the ocular surface. So external factors are microbiome, ambient agents, and individual factors are immune cells and hormones. We may have individual traits like breed, gender, age, lacrimal functional unit, precorneal tear film, the ocular surface, the eyelid, and we have environmental factors interfering with these individual traits. For example, we may have relative humidity decrease and an increase of tear evaporation. Concerning temperature, a decrease in temperature increases stimulation to the corneal cold fibers and reflex blinking. An increased temperature decreases reflex blinking and increases tear evaporation. The altitude, a decreased pressure at elevated altitude increases tear evaporation, but is exacerbated by a decreased relative humidity. And so we have an increase of tear film osmolarity. The draft, an increased air velocity around the ocular surface increases tear evaporation. And then also outdoor pollution, so air pollution and combustion products, but also indoor pollution, organic, volatile, emitted by cleaning and other products. So we must consider all these. And individual factors, so immune cells, the ocular surface associated lymphoid tissue that is composed of lacrimal glands associated lymphoid tissue, conjunctival associated lymphoid tissue, lacrimal drainage associated lymphoid tissue, and then hormones. Sex hormones have receptors in the lacrimal glands and meibomian glands. Androgens influence protein synthesis, immune response, and secretory functions. Androgens deficiency could lead to obstructive meibomian gland dysfunction. Growth hormone regulates meibomian gland size, morphology, and corneal epithelium migration. Thyroid hormone, corticosteroid, insulin, and melatonin have a role on ocular surface homeostasis. Well, we have considered the main function and effects of all components of the lacrimal functional unit. Let's now consider how to perform an advanced diagnostic approach because dry eye is not just Schirmer tear test. Diagnostic test must evaluate tear film stability, so the lipid layer, and we may perform meibography and interferometry to see the thickness and dynamics of the lipid layer. Must evaluate tear film evaporation, so the non-invasive breakup time and tear meniscus height. Must evaluate ocular surface wettability, so we can use interferometry or placido disc topography. Meibomian glands may be examined with non-contact infrared meibography, and you see the two instruments we are used to use here in the dogs and the cat. Infrared meibography allows to examine meibomian glands arrangement within the eyelids. We may use videos or shots to see all details of meibomian glands and their ductal openings, as you can see well here. A normal meibomian gland is approximately linear and 3 to 4 millimeter in length. 30 to 40 meibomian glands and the openings may be shown by non-contact infrared meibography in each eyelid. Meibomian glands number, the length, the size, more or less regular arrangement, and the dropout are easily examined in all animals. In most cases, the four eyelids examination with meibography lasts a few minutes. We may easily control the location of the meibomian glands, the ductal openings along the so-called gray line. Meibomian glands ductal openings may be occluded by cicatricial tissue, like in the horse here, and retroplaced, like in the dog, or capped by a dome of oil, like in the cat here. Meibomian gland dysfunction is a frequent finding in dogs with a history of long-lasting ocular surface disorder. In these pictures, we can see meibomian gland dysfunction with ductal opening retroplacement, occlusions, dilatation, dropout, and multiple distichia. Well, meibomian glands share strong similarities with the hair follicles of the cilia in embryologic development and structure. If germ cells fail to differentiate completely to meibomian glands, they become pilosebaceous units instead. And we have distichia, ectopic cilia that are easily examined by meibography. In this case, meibomian glands are normal, and there is an estimated loss of about 3%, but is nothing. And this is a case of meibomian gland dysfunction in a dog with sebaceous adenitis, with a loss of 70% in the upper eyelid and of 68% in the lower eyelid. Film interferometry. Oh, yes, let's talk about another advanced method of examination with film interferometry. We may use the OSAVET handheld ocular surface analyzer that is cable connected by a computer to a computer, or the TearVet a TearScope wireless connected to an iPad. The principles of thin film interferometry. Well, light striking a thin film is partially reflected and partially refracted at the top surface A. The refracted ray is partially refracted at the bottom surface. The two reflected rays have a phase difference and interfere to each other in a way that is proportional to the thickness of the lipid layer. And this is the origin of interference patterns. This is TFET analyzer, a wireless connected to the iPad, as you can see here very well, in a dog. And it is the same dog examined by OSAVET with OSAVET cable connected to the computer. Lipid layer patterns are evaluated to determine lipid layer thickness. We consider transparency, color, or texture. Color may be gray, may be whitish, bluish, pastel, or more intense, with or without waves. The pattern may appear more or less homogeneous, may appear dense with color interference fringes. The adopted frame and video grading scales are based on those available in human and veterinary literature. In practice, simplified free five-pattern grading scale are better used. And we can see horizontally and or vertically propagating waves at different speed and make some interesting deductions about the thin lipid layer composition. This is a frame grading scale I used to evaluate the interferometric findings. As you can see, there are 7 interference pattern grades from 15 to more than 100 nanometers, and a 0 grade to describe the hydrophobic plaque of lipid-contaminated mucus I found in several eyes. This is a video grading scale. Again, a 0 grade to describe lipid-contaminated mucus plaque, and 4 grades to show the different patterns and most of all thin lipid layer dynamics. In eyes with a lipid layer with a normal concentration of polar lipids, lipid spread on blinking is rapid and moves horizontally. In eyes with an altered lipid layer, the lid opening pulls up the aqueous phase first, followed by lipid spread in a vertically oriented pattern. And the evaluation of tear film dynamics must be done after a blink. When the lipid layer is thin or has an abnormal composition due to meibomian gland dysfunction or bacterial lipases, there is an increased tear film evaporation. Also contaminants, clumping of lipid mucins, hair, and loss of epithelial vertability increase tear film evaporation. This is a comparative evaluation of tear film evaporation in 2 dogs with the same shear material test and a lipid layer of different thickness at the start of anesthesia. At your left, evaporation is evident after 20 seconds due to the thin lipid layer. You see mucin filaments appearing on the ocular surface. With a thicker lipid layer on the right, the effect of a tear film evaporation is visible after 35-40 seconds. I examined with an interferometric study 146 eyes of 126 dogs with ocular surface disorder, and I saw about 22% of plaque of mucus, so grade 0 in these animals, and about 46% at the thin lipid layer, so grades 1, 2, with a transparent pattern, and about 26% had the denser lipid layer with grades 3, 4, 5, with waves, some colors, and only about 5% had the multicolor pattern, so grades 6 and 7 with waves. What does it mean? Grades 1 and 2 are a frequent finding, both with a normal tear film and in meibomian gland dysfunction cases. An increased tear film evaporation and an increased lacrimal secretion is a common cause of wet eye. Epithelial defect with loss of wettability determine the appearance of dry spot, and we have increased evaporation and decreased tear meniscus height and increased shim tear test with a wet eye. If we have a decreased shim tear test and we have an increased lipid layer in a normal functional meibomian glands, we see waves with limited and slow motion. So let's talk about non-invasive breakup time. Non-invasive breakup time is an important test to evaluate tear film stability. We add a Nibud grid to the instrument to project lines and concentric circles over the tear film lipid layer. In most cases, continuous eye and third eyelid movement to prevent Nibud evaluation. In any case, we can use the images to evaluate ocular surface topography, of course, like it's very visible in the bottom right picture. The right eye of this cat was normal at your left, and non-invasive bracket time was 17 and something more seconds. The left eye was wet with continuous blinking, and multiple dry spots were visible with interferometric examination, and non-invasive breakup time was about 0. Placido disc topography, where a Placido disc is inserted in the instrument to project concentric circles over the tear film lipid layer and evaluate their regular distribution. The top left video shows a regular topographic pattern. Topography, to the right, is very regular due to corneal defects in a bulldog with shimmer tear test 5. The Jack Russell underneath has a shear tear test of 2 to the right and 5 to the left. The right eye has a very altered tear film with musing aggregates and loss of ocular surface wettability, while the left eye has an apparently normal topography because the ocular surface maintains a pretty normal wettability. And then tear meniscus. Tear meniscus is another very useful test to be compared with shimmer tear test. Very important to compare to shimmer tear test. In evaporative dry eye cases, it's common to observe an increased shimmer tear test with a low tear meniscus due to intense tear film evaporation. Well, alternate use of interferometry is photography. The instrument to perform interferometry is useful also to make macro pictures to examine details of eyelids and ocular surface. We can make shots of meibomian glands ductal openings at the eyelid margin and document the occlusion. We can make shots of secreted lipids over the eyelid margin, like in these two pictures. With interferometric shots, we can easily document ectopic cilia. And the instrument lets make pictures of distichiae, and the loss of tear film for capillarity is evident sometimes, like at the bottom. Shots of conjunctival vessels are documented and better examined with this instrument. And we can also use the infrared light. Infrared light emitted with the meibography settings. Let's make shots to better document the presence of pigmented spots over the iris. An accurate diagnostic approach. Let's make a correct prognosis. Based on basic or advanced exam, I evaluated the prognosis with an ocular protection index. The basic ocular protection index considers eyelids, blinking, shimmer tear test, and fluorescein stains. 0, 1, or 2 points are assigned according to our examination. Eyelids may be inadequate to protect the eye, may have limited protection, or may be normal. We may observe absence of blinks, infrequent or normal blinking, and 3 levels of Schirmer-Tear test are considered, and fluorescein staining may be evident, faint, or absent. And with an ocular protection index value between 0 and 4, the prognosis, from my point of view, is negative. With an ocular protection index of 5, 6, the prognosis is good. With an ocular protection index of 7, 8, the prognosis is positive. The Advanced Ocular Protection Index considers meibography, interferometry, non-invasive breakup time, and ocular surface topography. 0, 1, or 2 points are assigned according to our examination as before. Meibomian glands dropout may be more than 50%, between 20 and 50%, or less than 20%. Lipid layer examination by interferometry may be inadequate, not fully protective, or normal. Non-invasive breakup time may be less than 10 seconds, between 10 and 15 seconds, or more than 15 seconds. And ocular surface topography may show severe alteration, mild alteration, or a normal pattern. With an ocular protection index value between 0 and 4, the prognosis is negative. With an ocular protection index of 5 to 6, the prognosis is guarded, and with an ocular protection index of 7, 8, the prognosis is positive. Well, we are close to the end. To diffuse information concerning meibography, interferometry, ocular surface topography, non-invasive breakup time, tear meniscus 8, SBM Systemi requested an atlas to be distributed for free to everyone interested to the topic, and it may be requested at info@sbmsystemi.com. I hope I increased your interest in the topic ocular surface and dry eye, to me one of the most important in the tyranny of ophthalmology. I wish everyone good health, success, and a great professional career. Thank you.